Alright, I believe this is the slide we left off on. We were on question 66, which is termination of transcription. If you watched that video that I posted before this one, it kind of summarizes the two processes of transcription and translation tied together. Translation did go kind of quick there at the end, but it kind of served the purpose. I'm going to go back and talk about termination again really quickly. Keep in mind, termination occurs when a stop codon is reached at the A site, which causes a water molecule to bond to the mRNA instead of an amino acid. And this causes the ribosome to fall apart and the mRNA to be released from the ribosome and the polypeptide or future protein to be released as well. So let's take a look at those processes right here. We can see a release factor, which is a water molecule, bonding to the stop codon, and let's check out what happens as a result of that. Keep in mind there's three stop codons, UAG, UAA, or UGA. Our last tRNA, it drops off its last amino acid here. It gets bumped out. Our amino acid chain, our polypeptide, gets bumped out. And now there's no more tRNAs in there. And we see everything falling apart. So that is the last stage of translation, that is termination. So now that we know what one ribosome does, let's talk about a polyribosome. This is a real scan of a polyribosome. We can see that there's ribosomes, looks like a dozen or so ribosomes on one single strand of mRNA. What this means is that there's a dozen ribosomes translating the exact same mRNA strand. So instead of just the mRNA strand being used to make one polypeptide, the same mRNA strand is being used to create a dozen polypeptides. So we're really cranking out polypeptides really fast, which is great because it's efficient with the time and efficient with the number of mRNAs. We don't need to make 12 mRNAs to create 12 polypeptides. We can use the same mRNA and translate it at the exact same time. And that's done by polyribosomes, which really is nothing more than just, as you look up here, a dozen or so ribosomes all working at the same time. So the combination of many ribosomes working together is a polyribosome, which simply breaks down to many ribosomes. So many ribosomes on the same strand of mRNA. One question you might have to try to answer that right now is, how do these two ribosomes or these two ribosomes not run into one another? They all work moving from the 5 to 3' direction. So all of these guys are all working in that same direction. And that looks like incoming is going this way, exiting is going this way, so they're all moving this direction, which makes sense. They're building onto the 3' end of the mRNA. So everything is moving this way, meaning that this guy cannot move this way to cause a problem. Leading up to questions 68 and 69, proteins or polypeptides that are created, they're not necessarily immediately ready for function. Sometimes they themselves need to be changed a little bit as well. So that's what we're going to be talking about here, is how do we modify those polypeptides, those proteins, to be functional after they have been created. So let's check that out. One thing that the video that I had posted prior to this did show was that after a protein had been created, it coiled up. Well, that's protein folding. That's one thing that did need to happen for a protein to become functional, is it needs to fold up into whatever shape it needs to take. So that happens after translation is done. Sometimes that might require a chaperonin. We had spoke about chaperonins previously, and if you're brain farting on what those are, they're little capsules that a protein can go inside of. They're hollow inside. They can go inside of it, kind of like an empty jar. So you put a lid on that jar, now it has a safe environment. The protein has a safe environment from outside forces so that it can fold into the shape that it needs to without fear of any interruption going on. So this would kind of be like you need to get away from your family, so you go to your car and you sit in your car and you close the door, and nothing can get to you, nothing can bug you, and you can kind of clear your mind. So that's kind of what chaperonins do. They provide a safe environment for the protein to take its final shape. So number 69 is talking about three different types of post-translational modifications that may be needed for the polypeptide, and here are those three right here. I'll explain those. Modification of amino acids. Sometimes an amino acid does need to be changed. Keep in mind that amino acids, which we looked at, I think way back in chapter 4 at this point, which seems like an ice age ago, they have functional groups. Sometimes we need to modify those or change those a little bit. So we might need to add on a sugar, a lipid, a phosphate to an amino acid. So we might actually need to change certain amino acids so that they function the way that they're supposed to. And it just happens. There's nothing that needs to be thought about or done. It's just something that it just happens. The second would be removal of one or more amino acids from the leading end of the polypeptide. This will be done by enzymes. Amino acids may be needed to be removed simply because they might serve kind of as a buffer, kind of like that 5' cap did for the mRNA. But the amino acid might not necessarily be needed for the protein's job, so if it's not needed, get rid of it. And that's exactly what happens. We'll remove an amino acid or so from some of the proteins, allowing that protein to become functional. And that usually happens from the leading end of the polypeptide, not necessarily the tail end. And the last part is, as with the case of insulin, insulin is produced in a really long polypeptide, but that really long polypeptide is not functional. So that polypeptide will be cut into smaller sections, allowing it to be functional, which is kind of strange. Why not just crank out the functional polypeptides to begin with? But that's not how it works. So you can almost think of this kind of like if you had a long piece of, I don't know, bread that's uncut. Like if you went to the bakery and bought bread and you had 4 friends over, well, it'd be pretty cool if all 4 of you were trying to munch on that same piece of bread. But that's not exactly sweet. So you would probably take a knife and you would cut that bread into 4 sections so that the 4 people could hold their own piece of bread and eat at the same time, rather than try to lock lips while you're eating some bread. So that's the case with insulin. It will take that polypeptide, it will chunk it up into smaller sections so that the smaller sections are now all their own insulin and all functional. So question 70 is asking you to use this image, looks like you need to label it, to explain how proteins are targeted for the ER. In other words, how do proteins get to the ER? The ER does need some proteins for its own personal function, and we can see that we have a ribosome delivering a protein to the ER. We have the lumen of the ER, or the lumen would be the hollow central portion of that particular organelle. So how this is done? Well, keep in mind that there's 2 populations of ribosomes in cells. There's the free ribosomes, which are floating around the cytosol, and there's bound ribosomes, which are attached to the ER. Most ribosomes synthesize proteins that are functional in the cytosol. Bound ribosomes make proteins of the endomembrane system, and these proteins are usually secreted from the cell. Keep in mind that the endomembrane system consists of several organelles, starting with the nucleus and ending with the vesicles, which are the little boat carriers which will carry a protein from the ER out to the cell membrane and remove it from the cell through the cell membrane, and that protein will go to some other organ or organelle or place in the body. Now, despite all mRNAs making their own unique and different polypeptide, the ribosomes themselves are all identical. They just read a different mRNA to make a different protein. But all ribosomes are pretty much the exact same. And they can sometimes switch even from free ribosomes to bound ribosomes. So they can be floating or they can be attached, and they can kind of go as needed. Polypeptide synthesis, it does always begin in the cytosol. And it usually will finish in the cytosol unless the polypeptide signals the ribosome to attach to the ER. So how do we signal for the polypeptide, which we see right here, the signal polypeptide? How do we use that to get into the protein? Well, we can see the signal polypeptide here, which is green on my screen. It is bound by a signal recognition particle, an SRP, to it. And then it's brought to a receptor on the surface of the ER. And this is where it kind of anchors to the ER, allowing that protein to finish translating. And as it translates, it goes straight into the ER lumen. This kind of reminds me of a bacteriophage infecting bacteria, if you recall that from chapter 16 when you looked at bacteriophages and infections with the marking of DNA. So more or less this SRP, the signal recognition particle, it's almost like making this boat anchor to this dock, and we can see the anchor going into the ER or whatnot, and it's popping off inside. So the signal peptide binds to an SRP, the SRP binds to a receptor protein, and that allows the ribosome to be bound to the ER. It pumps out the protein, and then it reaches its stop codon, which would be drawing in water. Everything falls apart. Ribosome's still out in the cytosol, so is the mRNA, but the protein's now inside of the ER. And this is how we get proteins to the ER.